Hello, my name is Prathyushasai Machineni. I am a master's student at the University of Houston, Clear Lake in Houston, Texas. I am currently working on my master's in biotechnology, and I am currently working on my thesis project. My project is about understanding pathways for microbial perchlorate reduction in a Mars analog environment. To start off with some background, we will be talking about the Mars and the analog environment first. So during the Phoenix lander mission, perchlorate was detected and observed at the Visitas Borealis landing site. Perchlorate was also evident in the rock-nest sand patches of the Gale crater, gathered by the Curiosity rover in 2012. Also, after the Viking lander mission's evaluation and detection of perchlorate, it can mean that it is globally distributed across Mars. Martian perchlorate can be an essential energy and oxygen research for future exploration. There are places on Earth that are analog environments. These include the Atacama Desert in Chile, Morocco, and the Pilot Valley Basin in Utah, USA. These environments have shown abundance of perchlorate. Examining different sites for biological and geological study can provide an interpretation of the biosignatures for comprehending the evolution of the Martian environment and possibly habitable life. So, for some basic on perchlorate. So, perchlorate, or ClO4- is a highly oxidizing anion with an oxidation state of +7 and a redox potential of 1.287 volts. Anthropogenic perchlorate in the environment results from various industries such as rocket propellants, pyrotechnics, fireworks, and the chemical industry. Perchlorate has been mainly studied as a drinking water contaminant because perchlorate can be a health threat to the thyroid by the inhibition of the uptake of iodide. I also have cited the papers along with the pictures just for a reference as well, and they will also be shown at the end of the presentation for further reading. So this picture shows the different pathways of perchlorate reduction pathways, including the enzymes and how it breaks down. So there are known different reduction pathways, which include canonical, symbiotic, and cryptic. The common perchlorate reduction pathway is canonical perchlorate reduction, which includes specific enzymes, PCRA, known as perchlorate reductase, and CLD, known as chlorate dismutase. The CLD enzyme is also present across chlorate-reducing bacteria and nitrate-oxidizing bacteria, which also uses Nixar, which is another type of specific enzyme, only specific to, or plausibly only specific to nitrate-oxidizing bacteria. The symbiotic pathway has so far only been observed in the laboratory and not in the environment. Cryptic perchlorate reduction is noted as an incomplete reduction because of the lack of the CLD enzyme, or chlorate dismutase, and it also has only been observed in the laboratory, just like symbiotic. The significance of cryptic perchlorate reduction in the environment is anticipated for further studies, as it could be an evolutionary step in the timeline of perchlorate. And as shown in this figure, we can see how it is broken down through PCRA and CLD. So the end products are chloride and oxygen. And this is the paper that the figure is referenced from for future reading. So a previous study by Lynch et al. 2019, a previous study has shown evidence of naturally occurring perchlorate, or NOP, found in the sediments of the Pilot Valley Basin, Utah. According to the study, site PV4 has shown a greater concentration of perchlorate in microbially induced sedimentary structures, also called MISS. PRMs, or perchlorate reducing microbes, have been found to be very phylogenetically diverse. It has been seen that perchlorate reducing microbes have been found to coexist with naturally occurring perchlorate, or NOP. From data of 16S rRNA gene sequences, both from sites PV3 and SLD124, shows what phylums, or which phylums, are dominant. PV3 showed proteobacteria and bacteroids are greater, and SLD124 shows that bacteroids and firmicutes are dominant. Known taxonomic groups for PRMs are Azospira, Halobacterium, and Pseudomonas. To date, over 40 isolates of perchlorate-reducing microorganisms have been studied and described. Perchlorate reducers are facultative anaerobes and can be inhibited by oxygen and nitrate. PRMs, or perchlorate-reducing microorganisms, have been commonly associated with the subclasses of alpha, beta, gamma, and epsilon of the Proteobacteria phylum. The main goal for this project is to identify which microorganisms metabolize naturally occurring perchlorate in Pilot Valley, why and how they are doing it. Another important question is, why is it important for a Mars analog environment? So a basic overview. The pathways that PRMs use are not entirely known. Previous work has shown that the canonical perchlorate reduction pathway is likely not the primary metabolic pathway. Nitrate-reducing microorganisms are also known to reduce perchlorate through nitrate reductase. A plausible pathway is through nitrate-reducing microbes due to the evidence of the NARG type reductase gene and the presence of nitrate-reducing microbial species. Enrichments have been already initiated by Dr. Kendall Lynch from 2019. The samples are housed in a minimal enrichment media to match the geochemistry of the Pilot Valley Basin. There are perchlorate samples, chlorate samples, nitrate sample, and blank samples that contain no oxyanions. We also have a kill control. The samples are interacted with an anaerobic chamber to maintain anoxic conditions or anaerobic conditions. Since working with the samples, a lot of protocol development has come way. We've been trying many ways to obtain better DNA yield and concentration. So the way we came up with it is we would obtain fresh sample from the culture bottle. The DNA samples are used for DNA extraction in triplicates plus a blank. The samples are then cleaned up and concentrated to obtain cleaner DNA and removing extra humic substances. These samples are quantified with a qubit fluorometer. qPCR is used to detect NARG and CLD. The standards are derived from a known perchlorate reducer, which is Dichloromonas agitata. The main goal is to identify the key enzymes in the pathways and identify the microorganisms. And we hope that NARG will show an increase and CLD will show an increase as well. Some further research. Through the continued study of perchlorate-reducing microbes, main perchlorate reduction pathways can be identified for metabolic energy. Genomic analysis and characterizations of PRMs through RNA sequencing can also be done. A perchlorate-based ecosystem model could be produced to study the implications of past, present, and future habitable zones on Mars. Finally, I would like to thank the University of Houston Clear Lake, the Lunar Planetary Institute, also known as LPI, USRA, and NASA for the opportunity and funding to work on this project. I would like to thank my PI, Dr. Kendall Lynch and Dr. Lori Santiago Vasquez, for their guidance, knowledge, and support on this project. I would also like to thank Jessica Lopez for her help in the lab. I would also like to thank AbGradCon for the opportunity to present at this conference. Here are the references that are mentioned in this presentation for further reading. Thank you for listening.